So during lecture, if you want to shout out, feel free to shout out, unmute yourself, ask questions. I don't mind that at all. You can also put it in the chat. I might notice your voice more than the chat box. So today, how'd that microscope lab go yesterday? It was
really long, but
it was long. Yeah, I had forgotten how long that was, and after I was going through it, I was wishing that I had gone through and picked like pieces and parts and maybe not had you do the whole thing. So I am sorry, that was definitely not for a 1 day. That would have been a good 2-day item. After we're done with our quiz, if you want to stay on the Zoom, if you had trouble getting into the gizmo, some of us struggled with that and we did some troubleshooting over the last 2 days. But if you still were unable to access that, stick around and I'll try to help you with that. So, so we're going to switch our order here. We're going to do today's topic, which is about cell membranes, and then we're going to roll back and take that quiz. That way, when you're done with the quiz, you can just take off for the rest of the day. Otherwise, you'd have to come back in 30 minutes, and then we would do our lecture. So we're going to swap that, and then. So what we have for our flex time is a worksheet. It's not, I promise, compared to the last one, it is not very long. It has a couple extra things to do, like read the textbook or do a Plix, like animation kind of thing. And the links are all hyperlinked on the document, or I also put them in as links in the assignment in Google Classroom, so you can access that either way. And then afterwards, we have a project. So the project is not due until next Friday, which is the last day of the term. This is a research project, and in the document, there is a list of organelles that have common genetic mutations that cause problems. So like mitochondrial disease, for example. So you'll pick a disorder that comes from one of the organelles, and you have lots of choices on the list. And then you'll have a research project and present a poster, a physical poster or a digital poster. If you wanted to do a PowerPoint, that's fine. A video, that's fine. This is going to count as an assessment, so a lot of you are looking for ways you can boost your grade. And most of us have high scores in the homework column. And since our grades are weighted, every item you do is only compared to that category. So if you already have a high percentage in the homework, you just need to keep getting that percentage so it doesn't change. But if you have a lower percentage in the test, like I said, you only compare what you're doing to that category. So if you have a 78% test average and you scored an 80% or higher, that's going to boost your overall grade. But if you scored 78, it would stay the same. And if you scored below that, it'll drop it. So because we're going to put this in the test category, that's a good chance for you to control the amount of points that you're earning in your test category, right? So you should be able to take pride in your work, accomplish all the tasks, turn this in, and get a high score. And that will help your grade, okay? So, so that's what that research piece is about. So that is in Google Classroom, and it is not due until next Friday. And because marking period is over, we can't take that late. Also, just a reminder, today's work, we're shooting for 3:30 in and out. Okay, so that's today's agenda. Do you have any questions over the agenda, the timeline, things that you need to do? All right, so then we're going to go to today's content, which is the cell membrane. Next week, we're going to talk about how things move through the cell membrane. So as you can see, the cell membrane is kind of complicated. It's got a lot of different structures in it. It serves a few functions. The obvious is that it's a boundary, right? So it regulates what enters and exits the cell, and that way it maintains homeostasis. So it keeps bad things out, good things in. It controls how much can move through and when it can move through. So like if you had sodium inside the cell, water is going to fill the cell and perhaps cause it to burst. So we want to keep the salt out, for example. So by keeping a stable state, the certain amount on both sides, you're maintaining homeostasis. I'm going to talk about another example of that in a little bit. It's a protective, so I said keep bad things out, right? And it supports or maintains the shape, integrity of the cell. It also surrounds many of your organelles individually. And then finally, it serves a purpose in communication. So these, we've talked about these before in the macromolecule chapter. These antennas, if you will, serve for cellular communication. Okay, so homeostasis, remember, is just maintaining a balance. So we keep the proper concentration of nutrients. If you need glucose, you need to take it in. Ions like calcium, potassium, sodium, magnesium, water, and then get rid of waste. So as the cells are going through their own metabolism, they're going to create metabolic waste like ammonia that you would need to get rid of. CO2 you need to get rid of. So it's selectively permeable, meaning it selects what can go through. So some things can and other things can't. Basically, if it fits, it ships. That's what I say. So the fluid mosaic model, fluid because it has fluid motion, all of these lipids in here, the yellow part, the tails are in constant motion. So that's why they refer to it as fluid. It's flexible. And it's made up of mostly lipids and proteins, but we'll also find a few other structures in there as well. So the lipid bilayer, this is what the phospholipid looks like, and this is going to be in your worksheet today too. So this top part here, this is the phosphate head. And these two tails here, these are the lipid tails. So again, go back to our macromolecules chapter when we talked about the triglycerides, right? So these are those fatty acids that would make up something like the triglyceride. So I can see the orientation where the phosphate heads are oriented to the outside of the membrane and the phospholipid tails, the lipid tails are facing inwards. Because this phosphate is polar, has a charge, it's attracted to water. Inside the cell, you have cytoplasm. Outside the cell, you have other extracellular fluids. So this phosphate head orientates itself toward the water. And then the lipids, like water and oil don't mix, right? You've seen that. So the lipids will orientate themselves away from the fluid, and that causes them to be on the inside of the membrane. So the fluid mosaic, fluid has to do with the motion. Mosaic has to do with the fact that when you look down on the membrane, it is a scattering of a number of different components. So if you've ever seen a mosaic piece of art, it's made of many different like ceramic pieces or porcelain pieces that go together to make art. Gaudi was big on that in Spain. So the mosaic is made of the phospholipid bilayer. You have several proteins. Some are for transport. They move things in and out of the cell. Receptors receive. So they're going to transmit information that they receive from outside the cell internally. So maybe you receive a signal that tells the cell, a muscle cell, to contract. Or maybe you receive a signal that tells a cell to make more white blood cells because there's an infection. So it receives that signal and then sends it inward to carry out the action. You have marker proteins that identify the cell. So they would identify whether it's a liver cell or a bone cell, for example. Cholesterol is in between each of those lipids, and it keeps them from stacking together or sticking together, so that maintains the fluidity. And then carbohydrates, those were the antennas you looked at. So those antennas serve in cell communication. So this is just a little animation, but I have to go through a few steps to get to it. there isn't a direct link, so I have to, oops, did I miss it? Here we go. So this is just showing you how the membrane has that fluid component. So you can see the lipids. Right now, all we're looking at is lipids. We're going to zoom in on those, and you can see that they're in motion. You can see that this portion here has a charge. Hydrophilic it is, water-loving. Here's a couple integral proteins, meaning they go all the way through the membrane. Peripheral proteins are only found on the edges, periphery, like your peripheral vision, only on the edges. And then the carbohydrates sticking up here, here's your cholesterol molecules. So you can see things within the membrane are moving, and then you can see the mosaic where it's a splattering of, if you will, a number of different components. Okay. So going on with those membrane proteins, many of them serve as enzymes. So remember, enzymes aid in chemical reactions, and under specific pH and temperature, they work best. So a lot of enzymes when we study photosynthesis and cellular respiration, we're going to learn about enzymes that are in the cell membrane. So they determine what can go through, they take part in chemical reactions, and then they act as identifiers. Like I have type A blood, so I have a certain glycoprotein, and sugar and proteins together, that identifies my cells as A blood. So if I were needing a transfusion and got A blood, my cells would read on the membrane of those red cells, would read that A sugar and know that you belong to me. But if I got a B blood, there's a different type of protein on the surface. This different marker protein identifies it as some other cell. My body would actually attack that because it does not belong in my blood. Okay? So that's a marker protein. These are transport proteins where they move items from one side to the other. This is a receptor protein. So a receptor protein is receiving signals. I gave you a couple examples. They have a very specific shape, both of these, that it's like an enzyme where they bind to only specific molecules. So two ways of communicating with our overall system, right, is the nervous system and the endocrine system. So we started our year with different types of systems. So the nervous system would release neurotransmitters. So a neurotransmitter is just a chemical messenger, and it binds with a receptor protein on the surface, and then that tells the cell what to do. So a good example with this is muscle cells. So the nervous system would release a protein that binds with a protein on the surface of your muscle cell, and that tells it to contract. Other examples would be hormones. And again, if that hormone binds to a receptor protein, a signal will be sent inside the cell telling it what to do. So it's kind of like that protein serves as a messenger. So here you can see some integral proteins that go all the way through. Here I see some molecules that are going through and other molecules that don't seem to be able to pass. Why do you think these two green things aren't able to pass? You can shout it out or put it in the chat. So what do you think? Why can these two molecules go through but not this? Any ideas? Out loud or in the chat box? Either one.
Well, the other ones are a lot smaller.
Thank you. That sounds like Jonathan, right?
Mm-hmm.
So yeah, if it fits, it can go through. So here you can see some pores. This is where it's selective. Not everything goes through. We'd say it's permeable if everything could go through. So only certain things. So these red blood cells, they're too big. They're not going to go through. But other things that your body needs, sodium, potassium, sugar, water, all those things are much smaller than a cell. So they're going to be able to go through. Viruses and bacteria, much smaller than our own cells, they could pass through some of our pores and infect our cells. So this is just another graphic of that. The blue ones you can see are much larger. They would not fit through these pores, so they're not going to be able to go through. when a substance moves through the membrane, we say that it's diffusing. So it's going to move from one side to the other side until both sides have approximately equal amounts. Diffusion, that's next week. So things that are smaller like water, carbon dioxide, oxygen, glucose, those things are all going to be able to move through our cells. Things that are larger, this is a good activity, but we're not going to do it right now. Things that are larger, like the food that we eat, we need that nutrient to get to all of our cells, right? And it travels through the bloodstream. But like the starch from my potato is way too big. So how am I going to get that starch through my cell membrane? I'm going to have to break it down. So again, remembering earlier chapters, I'm going to hydrolyze that starch into smaller substances using an enzyme until those macromolecules are small enough that they could pass through. Proteins are another item that's way too big to fit through your cell membrane. So either it's going to have to be transported with a transport protein, or like this, you'd have to hydrolyze it, digest it, break it down into its individual amino acids to move through. Okay, so in this picture here, do you think both the white circles and the black circles will move from one side to the other side? Do you think neither of them will move? Or do you think just the starch or just the glucose? You can shout it out loud. Shape is another good one, Gabby. You can shout it out loud or put it in the chat box. How will this membrane look after there is diffusion? Will both substances move or just one? Good, yeah. It looks like the starch molecule is much too large. I don't know much, but it looks like it's a little bigger than the opening. So the starch molecules won't be able to go through. The black ones are small. The glucose looks like it will be able to fit through. So if it fits, it ships. Okay, real quick, I'm going to give you 2 minutes. You have this self-check in your assignment. There are only 4 questions. So we're going to fill out the self-check, and that'll be the last task we have before our quiz. We need a good understanding of the cell membrane because next chapter we're going to start talking about movement through the cell membrane. They should be in your Google Classroom, linked with today. It says Friday Flex and Zoom both. Week 8, cell membranes, Friday, Zoom and Flex.
Okay, thank you.
And I believe it's the second one on the right, at least on my view. I don't know. It's under the slide set from today. You'll have the 2020 structures, and it's white with just 4 questions. What do you think? Why does the term what does the term selectively permeable mean? What molecules are able to easily pass through? Smaller molecules. Good. Okay. And what about those larger molecules?
Like they get digested into smaller monomers and then they can move through.
Good. And finally, how does the selective permeable cell membrane aid in homeostasis? Could you help me with the last question? Explain how the selective permeable membrane helps maintain homeostasis.
It only lets in the certain things it like needs for food and oxygen.
Good. Yeah. Perfect. And also getting rid of the things that would be bad for it. Okay. Any questions over today's topic? As we transition, is there any questions you wanted to clear up before our quiz?